Today we are going to talk about Krebs cycle. Krebs cycle is also called TCA cycle and it is also called citric acid cycle. So for practical purposes, Krebs cycle, TCA cycle or citric acid cycle is one and the same thing. Actually, this cycle is a special metabolic pathway. It's a very special type of metabolic pathway which runs in mitochondria, right? So first of all, I will do a little detail about mitochondria, then we'll go and discuss the Krebs cycle. Now, mitochondria are present in most of our cells. Most of our cells have mitochondria which act as mobile power plants, right? But there are few cells which don't have mitochondria. Can you tell me a single type of cell which does not have mitochondria? Erythrocytes. Very good. The red blood cells, they do not have mitochondria. Because red blood cells do not have mitochondria, they cannot run the Krebs cycle. Is that right? Because Krebs cycle enzymes which mediate the Krebs cycle pathway, they are present within the mitochondria, right? But most of our cells have mitochondria, and within the mitochondria this metabolic pathways run, right? So little detail about mitochondria. Mitochondria are intracellular organelles which are bound by the membrane, and they have double membrane. Look, suppose here is a mitochondria. This is the outer membrane of a mitochondria, and within it there are inner membrane of mitochondria. And inner membrane of the mitochondria has many folds. Inner membrane of the mitochondria have many folds, right? Black is the outer membrane and this is the inner membrane of the mitochondria, right? And this area which is between the two membranes, this area which is in between the outer and the inner membrane, this area is called, yes please, very good, intermembranous space. Intermembranous space. And this area which is internal to the inner membrane, right? This area is called, yes please, all the substances which are present inside the inner membrane, they are called matrix. Matrix. Right? Now, citric acid cycle or Krebs cycle runs within the mitochondria. This cycle runs within the mitochondria because the enzymes which mediate this pathway or the enzymes which run this cycle, they are concentrated in the mitochondrial matrix, right? Most of these enzymes which are running this metabolic pathway, right, they are present within the mitochondrial matrix or they are bound with the inner side of the inner membrane. Either these are within the matrix, these enzymes, or some of these enzymes are bound with the inner side of the inner mitochondrial membrane. So we can say Krebs cycle is a mitochondrial metabolic pathway. Question is that why this cycle is called cycle? Yes, question goes to Samad. Why this metabolic pathway is specially called cycle? For example, glycolysis pathway is not called cycle. Why this is called cycle? Yes. Because it continues Glycolysis can also continue. So, why this particular metabolic pathway is called a cycle? Yeah, in a way we can say that the final product of the pathway is absolutely identical with the initial substrate. Again, listen. Any metabolic pathway in our body in which the final product of the pathway is absolutely identical with the initial pathway, such metabolic pathways are called cycles, right? Now, what is the purpose of this cycle? We'll talk about that this Krebs cycle or citric acid cycle, what is the real purpose of it? What's it's doing there, right? Let me tell you. I will draw the cycle in a very simple way, right? I'm not putting all the detail of it. We'll discuss the detail later. Now, this metabolic pathway, number one function of this metabolic cycle is that it is concerned with the breakdown of acid, yes, acetyl moiety of acetyl-CoA, right? Acetyl-CoA. Now this is a compound, right, with how many carbons? Acetyl-CoA. This acetyl part has how many carbons? 2 carbons. It has only 2 carbons, right? Right, so double carbon unit. Basically, the purpose of whole this cycle is that acetyl-CoA should pass through this cycle and it should be catabolized while releasing energy. Now question is that from where the acetyl-CoA comes? Acetyl-CoA is the one of the breakdown product of, yes please, carbohydrates, lipids, yes, and amino acid, the protein, you can say it's one and the same thing, proteins. You know that proteins have carbon skeletons. Carbohydrates have carbon skeletons. Lipids have also carbon skeletons. When you will break down the lipids and eventually if you end up with double carbon skeleton, okay, I will make it here, double carbon skeleton, these are your friends. Now these double carbon skeleton, I don't know they're homo or hetero, but anyway they are together. So this double carbon unit, which is called acetyl-CoA, of course CoA is something special with it. This is the CoA part, CoA enzyme. Now this is the acetyl part of CoA. This acetyl part can be derived from the breakdown of carbon skeletons of lipid or by the breakdown of carbon skeletons of carbohydrates, or it can be derived from the breakdown of carbon skeletons of proteins, right? So it means that our major fuels in the body, right, major substances in the diet which we take like proteins and carbohydrates and lipids, all of them can be broken down into acetyl-CoA. Now acetyl-CoA is the major fuel. You know, this is energy producing cycle. This is energy producing cycle, and acetyl-CoA is the major fuel which enters into this pathway. Now what really happens when acetyl-CoA, right, moves through this? What are the major things? To get the energy, we have to remove this carbon. Let's come to a very simple thing. All of you must be knowing glucose. Can you tell me C6? Glucose, yes, H12O6. Is that right? Now, when you break it down into its component, it is what? You add the oxygen to it. And you end up with what? Yes, please. 6 carbon dioxide plus 6 water. But during this whole process, what really happened that you produce a lot of energy which is captured in the form of ATP. Now actually the purpose of this breaking glucose is that carbon and hydrogen bonds should be broken. The purpose of breaking glucose is to get the energy out of it. How you can get energy out of it? By breaking its carbon and hydrogen bonds. Now there's 6 carbon. Now you see that eventually if you get energy out of glucose, this 6 carbon should become the part of, yes, carbon dioxide. Is that right? So it means that these carbons should be puffed out as carbon dioxide, and these hydrogens and this oxygen plus this oxygen eventually should come out as water. Is that right? In previous lectures we were talking about glycolysis, and in the glycolysis we discussed that glucose, suppose this is glucose, how many carbons it has? 6 carbon, right? So let's suppose I make 6 carbon glucose, 1, 2, 3, 4, 5 and 6 carbon glucose. And we have seen that during the glycolysis process, eventually one glucose molecule break down into 3 carbon units, and these 3 carbon units are called, what are these called? Pyruvate. You know, glucose is 6 carbon. 2, 3, 4, 5, 6. During the glycolysis, right, the end product of aerobic glycolysis is that glucose eventually break down into the 6 carbon unit, eventually break down into 2 unit with how many carbons? 3 carbons, and these units are called pyruvate. Some energy is released up to that, but still within these bonds lot of energy is there, right? So these 3 carbon units which are called, yes, again, pyruvate, they should be broken into 2 carbon unit. They should be broken into 2 carbon unit from here, and also this should also break down into 2 carbon unit. It means that pyruvate, if you bring one carbon out, and these 2 carbon units are left behind, right? These are called acetyl moiety, and this acetyl part having a special enzyme with it, which is called, yes please, coenzyme, then this unit will be called acetyl-CoA. What I'm trying to put in your mind, that there are carbon skeletons in carbohydrate, in lipids, in proteins, and you have to break down carbon skeletons, is that right? And remove the hydrogens and get the energy. Am I clear? Now, as I told you, by glycolysis pathways, glucose can be broken down into pyruvate, and then from pyruvate we can bring one carbon out as with 2 oxygen. So what is it? Carbon dioxide. So from pyruvate you puff out carbon dioxide, and from pyruvate when one carbon come out, what is left behind? Acetyl unit, which you fuse with CoA and it becomes acetyl-CoA. Right, this was just one example that how glucose carbon skeleton is broken down into 3 carbon units of pyruvate and eventually 2 carbon units of acetyl-CoA. Am I clear? In the same way, later on we'll discuss that lipids carbon skeletons can also be broken down into 2 carbon units, which are called acetyl-CoA. In the same way, some of the proteins and amino acids carbon skeletons can be broken down into 2 carbon units, and those 2 carbon units are called, with coenzyme A, are called acetyl-CoA. So this is the acetyl-CoA. These are the 2 friends which will go into this cycle. Now when these 2 friends are passing through this cycle, right, what should happen? We have to release the carbon. Look, what is it coming out? Carbon dioxide. When the pathway is running, we have entered into pathway 2 carbon units. One carbon dioxide will be puffed out here. Then another carbon dioxide should be puffed out there. So it means that when acetyl-CoA is passing through this cycle, we are just talking about basic principles. We will go into detail of the reactions later. First, we concentrate on the main thing. When you put the 2 carbon unit, and as they are traveling through the pathway, their carbons are lost as carbon dioxide. So through this pathway, how many carbon dioxide molecules should be puffed out? 2 carbon dioxide molecules should be puffed out. Meanwhile, another thing, during this reaction, when NAD, when these 2 friends are passing through this pathway, some naughty people will come and take away their money. You see, this is energy rich. Acetyl-CoA is energy rich. These 2 friends come into this pathway. Not only they lose their heads, but they lose their energy also. And how do they lose their energy? The energy which is released, right, that is used in conversion of NAD into, yes, NADH. Now, how many NADH are produced by this pathway? Right? One is here, Then another NAD, okay, I will bring the NADH into more central position, NADH, because this is the one of the central product of this pathway. Then another NADH coming from here, right? Of course, NAD is converted into NADH. Then from here, another NAD will be converted into NADH. So in this pathway, you have lost 2 carbon dioxides and you have got how many NADH? 3. These are very, very energy rich molecules. And these NADH molecules will eventually go where? Very good. They will eventually go to electron transport chain, right? Let's suppose that is the chain, electron transport chain. This is also running within the mitochondria. This is a series of enzymes, and NADH will go to this pathway, right? Now when NADH will come to this pathway, what really happens? That its electrons will be accepted by NADH pathway, and what will be released? NAD. So basically the purpose of NADH is only to take the energy from the Krebs cycle to the electron transport chain. Is that right? The simple concept that when carbon skeletons are broken within the Krebs cycle, again listen very carefully. What Krebs cycle is doing? It is breaking down certain carbon skeletons and hydrogen bonds. When this carbon and hydrogen bonds are breaking down in the Krebs cycle, energy which is released, it is captured in many ways. One of the very important ways that energy which is released, that is captured in NADH. So as Krebs cycle is working, it is producing more, converting more and more NAD into, yes please, NADH. And NADH will be transferred to electron transport chain system, and it will release its electrons to electron transport chain, and its NAD will be released. Of course, it will come back again and it will be again utilized. So this NAD molecule is just like a mobile battery. That here the energy is packed into battery, and battery moves to the electron transport system, hand over the energy here, and it will become empty for refilling. For refilling, where it will go back to Krebs cycle. Is that right? Now, this is one way. Another way is some of the energy which is released, we'll see here is one substrate. When this substrate convert into next product, it releases energy, but that energy, attention please, in the pathway we'll see later, there's one substrate, when it convert into its product, it releases energy, but that energy is not enough to convert NAD into NADH, because more energy is required to convert NAD into NADH. So now we have to use a smaller battery. There is a smaller amount of energy released. We need smaller battery. The smaller mobile battery, molecular battery, which can take smaller amount of energy is, who will tell me? FAD. And it takes the energy and it converts into FADH2. And again this energy will be transferred there and it will come back, right? And in this way, what is happening? NADH is bringing the electrons here and protons there, and FADH is also bringing. So what is the purpose of Krebs cycle? We have learned up to now that Krebs cycle is accepting the carbon units, breaking them down, and energy which is released is being converted in the form of NADH and in the form of FADH2. Is that right? And these molecules, or we call it reduced NADH and FADH2, are called reduced coenzymes. These reduced coenzymes transfer the energy to electron transport system. Is that right? When energy is passing through this, in the end, what will happen? Who will tell me? What will happen is that, yes, oxygen will be added to this pathway in the end, and they are releasing protons here, and this is also releasing protons here. Protons will travel over this, meet with their beloved oxygen, and convert into H2O. You remember in that equation, when we are breaking glucose, we are ending up with carbon dioxide and water. Carbon dioxide is being puffed out here and water is being generated at the end. And during this, when oxygen is acting as a final oxidant to accept the electron and making water, the energy which is released here, that is used to convert ADP plus inorganic phosphate into ATP. You get it? Yes, please. What's your question? Yeah, look, the question is very interesting. Why the oxygen is the final oxidant in all these reactions? Thank God, because oxygen is abundantly available. Look, NAD is a good acceptor of electrons, but they have a limited supply. Do you think you can breathe in NAD or FADH? So they cannot be our final. What we do, that our metabolic systems, when they are breaking down the molecules, energy when it is released, initially this energy is loaded, filled into NAD system and FAD system. They get loaded with energy, and these energy loaded batteries have to come back, right, to be, so they will transport the, in a way, electrons or energy to electron transport chain, and we have to dump electrons somewhere. The best thing is oxygen, right? Oxygen is very easily available in the environment, and we can take it in our body, and the special transport mechanism to take oxygen from the lungs to the tissues, right? You know, everyone knows all those things. So because oxygen is readily available, so our metabolic pathways have developed in such a way during the evolution, they dump their electrons on oxygen, and oxygen get reactive, and that oxygen, reactive oxygen loaded with electrons, you know, it becomes very in search of its friends and react with hydrogens. And in this way, when we get carbon skeleton in our body, let me tell you, when we talk about glucose, it is something like this. Is that right? Am I clear? There are, now actually what we do when we are breaking these bonds and getting the energy out of these bonds, we have to deposit carbon somewhere. We don't want that carbon is deposited in our body like coal. We want carbon to get out of our body. The best way to get the carbon out of body is fuse the carbon with oxygen, and you get a gas which is called carbon dioxide, and our body has developed beautiful system to puff out the carbon dioxide. Secondly, now we are left with hydroxyls, and this, what we will do? Water. And you know how to get the water out of body. So actually this is, I mean, even small kid knows. Right, now listen. So what I'm talking about, the whole Krebs cycle story is about this, that this is a metabolic cycle which is very, very efficient in breaking different type of carbon skeletons, releasing the energy and filling the energy temporarily into NADH and temporarily into FAD, and then they will take that electrons to the electron transport chain and regenerate their empty packs, right? And electrons will travel over that, activate the oxygen, and oxygen will react with hydrogens, right? Meanwhile, the energy, I will explain into detail. There will be lectures on electron transport chain and oxidative phosphorylation, that there's a process of oxidation in which ADP is phosphorylated. So it is oxidative phosphorylation. Why we call this process the last process in electron transport chain is called oxidative phosphorylation? Because we are doing phosphorylation of ADP. We are adding phosphate to ADP. So this is phosphorylation. But if there is no oxygen, can we get this phosphorylation? No. So this is called oxidative phosphorylation. Am I clear? No problem. Now look at another thing. Some energy is directly produced. Look here. There is a substrate here. When it will convert into its product, some GDP is converted into GTP, and this GTP can go back while converting, yes, ADP into ATP. So it means when cycle is running, a little ATP is directly produced. Now listen carefully. What is the purpose of this cycle? It's very clear. You pump the cycle with carbon units, And when carbon units of breaking, energy is being released, and cell does not let that energy to be destroyed. That energy is captured. Most of that energy is utilized in converting NAD into NADH. Some of this energy is utilized in converting FAD into FADH2. Is that right? So when what are these? When during this cycle, carbon units which are moving through that, when they are getting oxidized, right, then NADH and FADH are getting reduced. Because they are getting reduced, they are also called reduction equivalents. So we say one of the major purpose of Krebs cycle is take up the carbon skeleton, and it loves to take acetyl-CoA, right? And it breaks it down. And as substances are moving through this, they undergo decarboxylation reactions. Which are these reactions? Decarboxylation reaction, because carbon dioxide is released. So during this pathway, there must be decarboxylation reaction. Plus, there can be dehydrogenation reactions. It's very easy to understand whole this pathway is about what? This pathway is about decarboxylations and dehydrogenations. Decarboxylation help us to puff out, blow out carbon dioxide, and dehydrogenation help us to transfer the hydrogens and electrons to coenzymes. Am I clear? So one way to define this pathway is that Krebs cycle is a special metabolic pathway in which you pump in acetyl-CoA or other carbon skeletons, right? And these substrates pass through sequence of decarboxylation reactions and dehydrogenation reactions, and carbon dioxide is blown out from the cycle, and NADH and FADH2 is produced, which will transfer the electron to the electron transport chain. Eventually those electrons will move on the electron transport chain and result into oxidative phosphorylation of ADP into ATP. Is that clear? And a little energy is directly produced. That I told you, one GDP is directly converted into GTP. And then GTP, guanosine triphosphate, gives its phosphate to ADP to make ATP. So in this pathway, a little ATP is directly produced. Now this production of little ATP is not involving electron transport chain, or is it involving? This ATP is directly produced. This is not involving the, yes, electron transport chain system, and it does not involve oxidative phosphorylation. Look, this step, when this substance convert into next substance, I will tell into detail every substance later, but first we just develop a main idea. When this substance is converted into next, so there's one metabolic step during the Krebs cycle in which directly one molecule of ATP is produced. This production of ATP does not involve electron transport chain. This production of ATP does not involve oxidative phosphorylation. So we call it substrate level phosphorylation. That one substrate went to the product, and at the same time, simultaneously and directly, it phosphorylated the GDP into GTP and resulting into phosphorylation of ADP into ATP. This step is called substrate level phosphorylation. Now, I told you this is you are getting the cache energy. When you get ATP or GTP from a metabolic pathway, you are getting cache energy. And NADH and FADH are check. They are not cache. You have to take NADH and you have to take FADH2 to the bank of electron transport system. Electron transport system bank then gives you the cache, this ATP. Is that right? Again, listen. Energy which is produced by Krebs cycle, some energy is coming as cache, some energy is coming out as check. Cache is a small amount is cache, as during one cycle, one ATP is produced. And 3 checks of NADH are given to you, and 1 check of FADH2. You take these checks to electron transport chain and you will get the cache here. Now, what is the worth of one NADH check? NADH check. This is equal to how many ATPs? Yes, we have discussed previously that one NADH, right, when it interacts with electron transport chain, it end up into 3 ATP molecules. Is that right? And one FADH2 produces how much ATP? More than NADH or less than NADH? Less. It produces 2 ATP. Now look at the energy production through this pathway. It's so easy to understand. How many NADH are there, my friends? 3. So when this 3, during one cycle of Krebs cycle, 3 NADH are produced. When these 3 NADH will pass through the system, how many ATPs will be generated? 9 ATP. So 9 ATP are generated when 3 NADH are going over there. Plus 1, what is this? FADH2 is passing through that. How much ATP is produced? 2 ATP. And 1 ATP was produced by substrate level phosphorylation. So how many total ATPs are produced by one Krebs cycle, one swing? 12 ATPs. Am I clear? Again, energy production. For example, we get one acetyl-CoA molecule in. Now you imagine, this is a cycle. This is like a, and to run this cycle, we have to add something here. When you put one acetyl-CoA in the cycle, attention please, when you put one acetyl-CoA in the cycle, cycle will spin. Cycle will spin, and during one rotation, how many ATPs are produced? 1 ATP is directly produced, 3 NADH, which are equivalent to 9 ATPs if oxygen is available. Because if there is no oxygen available, then the trouble will be there. You know why? Let's suppose there is no oxygen. You develop severe hypoxia. And if there is no oxygen, then do you think, just a minute, when there is no oxygen, do you think this oxidative phosphorylation pathway will run? No. Do you think NADH can be converted to NAD? If there is no oxygen, can you convert FADH2 into FAD? No. So it means if there is no oxygen, look at it. If there is no oxygen, electron transport chain stop. When electron transport chain stop, NADH2 and FADH2 are accumulated. Cell develop accumulation of NADH2 and cell develop accumulation of FADH2. When these two are accumulated, right, this is the signal that when these two are accumulated, what will happen to this? They will be down. And if there is no NAD and there is no FAD, can the cycle run? No. Am I clear? Whenever there is no oxygen here, again, you cannot reproduce NAD from NADH, and you cannot reproduce FAD from FADH2. And when cycle develop deficiency of NAD and develop a deficiency of FAD, can it run? It means that even though oxygen is not directly utilized here, but this pathway still runs when oxygen is present in the mitochondrion for electron transport chain. Because this pathway is dependent on oxygen, we call it aerobic pathway. We call it aerobic metabolic pathway. Let me tell you again. Suppose there is no oxygen. If there is no oxygen, whatever NADH is produced and whatever FADH is produced, right, it cannot be utilized into this pathway because oxygen is not there. And NAD and FAD cannot be produced. And if these two are not there, pathway cannot run. It will stop, stand still. Am I clear? So in severely hypoxic tissue, Krebs cycle or citric acid cycle or TCA cycle stops. No problem up to this? Now you imagine if someone asked you a question that one molecule of glucose has gone into a cell, one molecule of glucose has gone into a cell, how much ATP it will produce in the presence of oxygen? Listen, question is, one molecule of glucose has gone into cell, metabolic pathway of the cell are intact. Metabolic pathways of the cell are intact. One single molecule of glucose first will break down through glycolysis into pyruvate. then pyruvate will convert into acetyl-CoA, then acetyl-CoA will spin through Krebs cycle. Through all this pathway, how much ATP is produced? Raise your hands, how many of you can calculate? Vyagne, right. Total in energy production. Just a minute. I will help you to understand. From glucose forward, 1, 2, 3, 4, 5, 6. This is glucose. From glucose, glucose convert into what? Glucose convert initially into glycolysis pathway into glucose 6 phosphate. Glucose 6 phosphate goes into fructose 6 phosphate. You have to watch the videos of glycolysis to understand it. Fructose 6 pathway is converted into fructose 1,6 bis phosphate. Fructose 1,6 bis phosphate is broken down into glycerol, aldehyde, 3 phosphate and DAHP, dihydroacetone phosphate, which is also converted into glycerol aldehyde 3 phosphate. Then glycerol aldehyde 3 phosphate is converted into 1,3 what? Glycerol aldehyde 3 phosphate is converted into 1,3 bis phosphoglycerate. So 1,3 bis phosphoglycerate. Then it goes into 3 phosphoglycerate. Then into 2 phosphoglycerate. Then into phosphoenol pyruvate, and then it will go into pyruvate, and then into acetyl-CoA and acetyl-CoA. Now this acetyl-CoA will spin through 2 cycles, one cycle of this and one cycle of this. Now we'll see the energy production. This is a very important concept in biochemistry, that we have taken one molecule of glucose and abundant oxygen is available, how much energy will be produced. Here, this was glucose into glucose 6 phosphate. It means 1, 2, 3, 4, 5, 6 glucose with 1 phosphate. It means this phosphorylation is done by investing 1 ATP. Is that right? Then what we do? We have added 1 phosphate here. Then fructose 6 phosphate is simply isomerization. Then fructose 1,6 bis phosphate. This is how many carbon? 1, 2, 3, 4, 5, 6. But carbon at one position, phosphate at one position and phosphate at 6 position. So it is double phosphates. Phosphate at carbon number 1 and phosphate at carbon number 6. Right? Now listen carefully. It means we have added one more phosphorylation. From where the phosphate comes from? ATP. Up to conversion of glucose, up to fructose 1,6 bis phosphate, we have invested how much ATP? 2 molecules of ATP. We have pumped into system, right? Now, this will break down into 3 carbon units, right? This is 3 carbon and this is also 3 carbons, and these carbons are having 1 phosphate here and 1 phosphate here. Is that right? If you remember the early lecture, when you convert glycerol aldehyde 3 phosphate into 1,3 bis phosphoglycerate, what really happens that you get what thing out? NADH. That is the point which is very, very important here, that here you will get 1 NADH. And remember, if 1 NADH goes through electron transport chain in the presence of oxygen, how much ATPs will produce? NADH will produce 3 ATPs, NADH. Now, from here up to here was investment phase. We invested the ATP in. From here we got 1 NADH here and 1 NADH here out. Is that right? Of course, this NADH will also move to the where? Here and produce ATP here. Are you understanding? So this is what, 2 packs of NADH we get. During this, inorganic phosphate bind over here. So it becomes, now look, this is 3 unit carbon still, but how many phosphorylations it has? Phosphorylation at 1 position and phosphorylation at 3 position. So 1 position phosphorylation and 3. 1 position phosphorylation and 3. There are 4 phosphate, 2 phosphates here, 2 phosphates here. Of course, when we will remove these phosphates and add to ADP, how many ATPs will end up? 2 from this side and 2 from this side. So what really happens? The 1,3-bis phosphoglycerate convert into 3-phosphoglycerate, and it means third phosphate is there, 1 phosphate has been lost. It was 3 phosphate, 1 phosphate is lost, and that phosphate is lost in production of what? ATP. So again, ATP is produced here and here, right? Now this is 3-phosphoglycerate, then 2-phosphoglycerate, then phosphoenol pyruvate. Phosphoenol pyruvate is again, listen, how many carbons are still there? 1, 2 and 3. Look, this is all 3 carbon unit. We are just modifying the molecules so that we can get another phosphate out. Our eyes are on that phosphate. So here 1 phosphate more will go out, when, right? This is 1 phosphate, but when this phosphoenol pyruvate convert into pyruvate, that 1 phosphate is lost and pyruvate is just 3 carbon unit. And when this phosphate goes out, you get what? 1 more ATP, because ADP will come and take the phosphate and ATP. Is that right? We will not talk about enzymes. We will discuss them into detail in the video lectures of glycolysis. I'm just trying to make a concept in your mind that how energy is extracted when glucose molecule is undergoing different modifications and we are breaking down its carbon and hydrogen units, right? And how energy is captured in which forms. Listen. Now, acetyl-CoA, this is going into acetyl-CoA, and this pyruvate converting to acetyl-CoA. Here also pyruvate is converting into, yes please, acetyl-CoA. Is that right? And then this will pass through this cycle. Now listen, up to now in aerobic glycolysis, because aerobic glycolysis means oxygen is available in abundant amount, and if there's a lot of oxygen available, the cycle should be running, and if the cycle is running, pyruvate will go down into acetyl-CoA. But if oxygen is not there, pyruvate will go on the sideways into lactate. Now listen, if oxygen is available, pathway will go down. But let's talk about up to now what we have earned. We have invested 2 ATPs and we got 2 NADH and we got how many ATPs? 4 ATPs. So net ATP production up to this point is up to pyruvate. Just a minute, I will put a line here. The business, we have run our business from here up to here, and we got 2 ATP invested, right, and 4 ATP generated. 4 ATP generated minus 2 ATP investment. Like a good businessman, getting your investment out, right? So what is the net production of ATPs? 2 ATPs up to now. Plus how many NADH we have produced? 2 NADH. And of course NADH will run through that. In the end it will produce how many ATPs? 6 ATP. So total, how much ATPs you have generated up to now? 2 NADH was 6 ATP, because 1 NADH produced 3. So 8 ATP molecules. Now what we have seen up to now, that in situation when oxygen is abundantly available, then glucose is broken down up to pyruvate in glycolysis pathway, and net production of ATP is 8 ATP. Is that clear? Now we move forward. Yes, pyruvate is 3 carbon unit. Krebs cycle acetyl-CoA is how many carbon unit? 2. So it means this is going to be 2 carbon unit, isn't it? It means when you convert pyruvate into acetyl-CoA, we have to get 1 carbon unit out. So this is our production of what? Carbon dioxide. And here's your beautiful production of carbon dioxide. So what really happens when pyruvate is converted into acetyl-CoA? You know, this line is telling the boundary between, okay, that we'll talk later. Pyruvate is converted into acetyl-CoA. What really happens that carbon dioxide is puffed out. During this process, because this enzyme complex, listen carefully, the enzyme complex which convert pyruvate into acetyl-CoA has decarboxylase function as well as it has dehydrogenase function. Because look, when 3 carbon unit pyruvate and we cut off 1 carbon, energy will be released or not? Energy will be released. We can add that energy, immediately pump that energy into NAD. So what really happens at this step, 1 NAD is produced, NADH, right? NADH. Because we have removed 1 carbon from pyruvate and converted pyruvate into acetyl, right? CoA, and 1 carbon dioxide molecule is puffed out, blown out of this reaction, plus 1 NAD is converted into NADH. So it means between the glycolysis and after the glycolysis and before the Krebs cycle start, because end of the glycolysis is at pyruvate and acetyl-CoA is going to be the part of the Krebs cycle. But this pathway, conversion of 3 unit carbon pyruvate into 2 unit acetyl moiety associated with CoA needs the reaction of decarboxylation and also is coupled with dehydrogenation, dehydrogenase activity. Now this NADH will produce how many? I will put them star NADH. So you get 2 star NADH will produce how many? 6 ATP. But this ATP will be of course only produced when there is oxygen. Right? Now the game will really enter into what? Krebs cycle. In the Krebs cycle, look at this Krebs cycle. In the Krebs cycle, now you have to tell me how many carbon dioxides are blown out. 2 carbon dioxide here, and how many carbon dioxide here also? 2. By the way, look at the beauty. It started with how many carbons? Are you sure? 6? Every carbon will blow out as carbon dioxide. Now how many carbon dioxide we have produced? 1 when pyruvate converted into acetyl-CoA. Then during the Krebs cycle you have produced 2 more carbon dioxide. How many carbon dioxide through this one pathway? 3. And another pathway? 3. Total how many carbon dioxide? 6. Equation is getting balanced in your mind that while we are breaking down the glucose, we get 6 carbon dioxide. You know exactly at what position. 2, right? 2 carbon dioxide just before the Krebs cycle start and 2 carbon dioxide from each Krebs cycle revolution. Any question up to this? But you know already that from here also 1 NAD will come, NADH, from here also 1 NADH, and from here also. So how many NADH are here? 3. Okay, this is 3 NADH. Here also NADH, NADH, and later on we'll learn one more. So here is also 3 NADH. So these 3 NADH are the part of the Krebs cycle. So Krebs cycle NADH is equal to 3 NADH is equal to how much? 9 ATP. 9 ATP? No confusion? Anyone who is confused? Okay. Then we have said that there was direct phosphorylation. So one ATP will be produced on this side, listen, and one ATP will be produced on this side. But again, we did a mistake here. Yeah, you are yourself intelligent. People have their friends who are intelligent. Yeah. So what I'm talking about here should be 3 NADH from one acetyl-CoA, and of course from one glucose molecule there were 2 pyruvate and 2 acetyl-CoA plus 3 more NADH. So total how many NADH are produced? 6 and 18 ATP from here. Then there are one ATP directly produced and directly produced. So substrate level phosphorylations, how many ATPs? Right, 2 ATP. Then there are, you remember there was your friend FADH2, DH2 and FADH2. So 2 FADH in when both of them pass through that. So 4 ATP. Now let's calculate. My math is not so good. Look, 8 up to glycolysis, when one molecule of glucose is passing through up to pyruvate, it produces 8. When 2 pyruvate convert into 2 acetyl-CoA, 6 more. So yes, we'll put it here. So they become 14, right? When 2 acetyl-CoA runs through twice through the cycle, 6 NADH are produced. Each cycle produces 3, and double 2 acetyl-CoA produce 6 NADH, 18. 14 and 18 is how much? 14 plus 18 is, are you sure? 32. Then 2 are directly produced, plus 34 plus 4 from FADH2. So it becomes 38. So this is how energy is produced from breakdown of glucose. Yeah. One molecule of glucose, if abundant oxygen is present and mitochondria are working, then one molecule of glucose will break down into 6 molecules of carbon dioxide plus 6 molecules of water plus convert 38 molecule of ADP into ATP. Is that right? With that, no, that initial investment we brought out. Otherwise, if that's the net value. Yeah. Now what I will do, I will take a test of you. Attention please. I will remove all of it and I will take a test of you here. And you will tell me how the ATP is coming out. I will not write into detail. Now you have to calculate the energy input and output of one glycolysis. Look, there is one glucose, okay. One glucose going into how many pyruvate, my friends? Pyruvate and pyruvate, right? Up to this, what happened? Now you will tell me, hurry up. How much in investment? 2 ATPs, excellent. We put it in. Rather, before the pyruvate, we go to glycerol aldehyde 3-phosphate. And glycerol aldehyde through one step, aldehyde 3-phosphate. Then glycerol aldehyde 3-phosphate eventually come into pyruvate. So what will come out from here? 1 NADH, very good. Also from here 1 NADH. And how many ATPs? ATP, and this ATP is not directly involving the electron transport chain and oxidative phosphorylation. So this ATP is produced by substrate level phosphorylation. That's great. ATP and ATP. So the net business of glycolysis, right? The net business of glycolysis, you put 2 ATP and get 4 ATP. So how many net ATP you get? 2. So you have 2 ATPs profit plus in profit you have how many NADH? 2 NADH. This is the net business balance sheet at the end of glycolysis. Then the junction of glycolysis with Krebs cycle is the point where pyruvate convert into, yes my friends, acetyl-CoA. To convert into acetyl-CoA. Now you have to tell me, at this level, I'm talking only about energy thing. There are other things also happening. Very good. NADH and another NADH. So it means glucose to pyruvate. This was our net profit. And at this juncture, from pyruvate up to acetyl-CoA, we got 2 NADH. Is that right? Now we come to the cycle. When the Krebs cycle runs, yes please, how many NADH are produced? 3 NADH, right? 3 NADH. Later on we'll go detail of reaction. I will tell you which reactions are producing, right? 3 NADH. Plus you are getting what? 1 FADH2. FADH2. Plus you are getting what? Directly 1 GTP or ATP, because GTP if you get, you can convert that into ATP. Right. Now Krebs cycle business. Your glucose breakdown business can be divided into 3 segments. First part of the business is aerobic glycolysis. Second is the junction of glycolysis with Krebs cycle where pyruvate breakdown to acetyl-CoA. And last part of the business is when acetyl-CoA runs through the cycle. Right. What is the energy spent out? How many ATPs? 2 ATPs directly. How many NADH? 6 NADH. How many FADH2 by both sides? 2 FADH2. Is that right? Now if someone asks you that when one molecule of glucose and its derivative pass through the Krebs cycle, what is the total energy released? 2 ATP will be equal to 2 ATP. And 6 NADH will be, yes, 18 ATP. And this will be 4 ATP. Yes, what is the grand total here? Only about the Krebs cycle, 24 ATP. Now listen carefully. Something which confuses the students during MCQs and studying the book. If in the book they are referring to one Krebs cycle, it means they are talking about one acetyl-CoA and one pyruvate. Then how many? 12. Because 9 from here, 10th is this, 2 ATP from here. So one revolution of the Krebs cycle produces finally 12 ATPs. But one breakdown of glucose results into 2 cycles, so it produces 24 ATPs. 24 ATPs you get here. These NADH at the junction will get how many ATPs? 6 ATP. So those Krebs cycle 24 plus pyruvate to acetyl-CoA 6. How much it becomes? 24, 30. And how much you got here? 32. And these equal to 6, 38. Any question up to this? No problem. So we can say when glucose is breaking down through the glycolysis and eventually through the Krebs cycle in the presence of abundant oxygen, when NADH and FADH produced through these pathways can be properly, you can say, used up by electron transport chain and can result into appropriate final oxidative phosphorylation, then one molecule of glucose will break down, will eventually release 38 molecules of ATP. Now listen, one point which I want to make it clear, that sometimes what happen, oxygen is not available. If oxygen is not available, this pathway can run up to here. And then in the absence of oxygen, pyruvate will convert into lactate. And this pyruvate will also convert into lactate, lactate dehydrogenase. But during this process, this NADH will be used and back into NAD. And this NADH will be broken down here back into NAD, because look why it is happening. Listen very carefully. If there is no oxygen available, start from here. No oxygen available, ADP cannot be converted into ATP. This oxygen is not there. If oxygen is not there, electron transport chain cannot run. When electron transport chain cannot run, NADH and FADH cannot be broken down into NAD and FAD, right? When NAD is not available, can pathways run forward? No. So due to this reason, when oxygen is not available, this NADH will accumulate and it cannot run forward. Then, but to move this point up to this, we need NAD to convert into NADH. So what really happens, whatever NADH is produced from here, right, while pyruvate is converted into lactate, NADH is converted back into NAD. So it means that in this pathway, if there is no oxygen available, NADH which is produced at this step is lost at this step. So only 2 ATP are invested and 4 ATP are gained, and net production is only 2 ATP. Why I am stressing it? What I want to put in your mind, when there is no oxygen available, one glucose will break down into, eventually in anaerobic circumstances, one glucose will break down up to 2 lactate, and there are only 2 ATPs. Again listen, when there is no oxygen to the cell, right, under those circumstances, what really happens is that Krebs cycle does not work at all in the presence of oxygen. Why? Because NADH and FADH2 which are accumulated, they inhibit the Krebs cycle, right? And NAD or FAD are not available. Then whatever NADH is produced in this pathway, that will convert into NAD by conversion of pyruvate to lactate. So what really happens that when oxygen is not available, when there is anaerobic glycolysis, the whole process of anaerobic glycolysis produces only 2 molecules of ATP per glucose. But if you supply the oxygen and mitochondria start working, then 1 molecule of glucose will produce 38 ATP. So this must tell to you clearly that under anaerobic glycolysis only releases very small amount of energy from the glucose, and aerobic glycolysis really produces a massive amount of energy from the glucose. Is that clear? Let's have a break.